We will start our last set of case studies looking at this funky little situation. The DPDX team received a worm-like object collected from a child's diaper. How fun would that be? Grossly, and they don't mean grossly, they mean like macroscopically, but also grossly, the object was white, semi-flattened, and measured about 4 millimeters by 2 millimeters. With no discernible external features, it was teased open and its internal contents were examined microscopically. As you can see here, figures A and B show it was observed from the teased preparation of the object. And you can kind of see how big they are. This is a unique situation with eggs, so I hope that it kind of jumps out at you what it is. If you need to take a moment to go and look, go ahead and look. Pause me. Otherwise, we're going to talk about these weird egg packets that hold between 5 and 30 eggs that come from the organism known as the dog tapeworm Diplydium caninum. And Diplydium caninum kind of looks like this as when it's an adult, I guess we would say. And what's unique about it is that its proglottids are long and skinny, and they are kind of barrel or pumpkin seed shaped. This one also has some polka dots in it, but you can definitely see the size and shape between this one and Diphyllobothrium latum are quite striking. It's really not the same. And we can look at the Scolex here. It does have 4 suckers. It does have a retractable armored rostellum. It sounds like something that some sort of funny superhero would have, right? An armored retractable rostellum. But this is known as the dog tapeworm, although it's not only in dogs, animals in general. Here's a cute little cat that can transmit it. And we're going to start at number 1 at the bottom, where gravid proglottids are passed intact in the feces or emerge from the perianal region of either animal or human host. I actually think that these have some capability of kind of wiggling themselves out of the perianal region, which is wild. Anyway, these proglottids that have egg packets in them will basically, these are egg bombs. These are the ones that are like true egg bombs because it's the proglottid and then it has all these packets of eggs. And the packets of eggs are held together by this outer membrane, which you can kind of see here. And inside you have all these little eggs. Oftentimes this membrane will disintegrate. So when you look at them under the microscope, you don't actually see the membrane, but you see the eggs themselves. Now, what'll happen then is a little larval stage of a flea will eat the eggs that are in here. And once that happens, the eggs will develop into that oncosphere, which is sort of that creepy little early stage that will hatch. And then it penetrates the intestinal wall of the flea larva and develop into cysticercoid larva in the body cavity of the flea. And then it will continue as the flea develops, so does this little parasite, and the adult flea will harbor the infective cysticercoid. So it doesn't do that much between larval and adult flea stages, but it is still the infective stage. Now, also the weird thing, the way you're going to get this is you have to eat a flea. And I'm willing to bet that cats nor dogs nor humans will willingly or knowingly eat fleas, but I'm sure they could hop in your mouth at night when you're sleeping. So when you get the flea into your body, then it's kind of like all the other ones that cysticercoid larva will emerge. It will attach. It will develop into an adult. You can see the Scolex here is very pointy. This is one thing. It's like way pointier than Taenia solium and saginata, and the head is a little bit, I don't know, it's like rounder, I suppose. And this is often, as you read here, humans, now normally children, acquire the infection by ingesting the flea. So imagine like a kid hugging a dog that's covered in fleas with their mouths open, right? And probably kissing them. Like, you can see that happening. And then what'll happen is we kind of take the place of the cat or the dog or whatever it is, and we will pass out these little proglottids in our feces. And of course, the cycle continues. Give me one moment to just check here. I really thought that one thing to note is that these are double pored. They're called double pored tapeworms because they have 2 genital pores on either side, which is kind of important. And it's not I mean, although proglottids can be found in the stool, it's not as common with the other organisms as it is for this one, especially the little ones, right? The little ones for sure are not going to be able to, like, lose a chunk of their body because that is their whole body. So Diplodium caninum, right? This is the dog tapeworm, the little kid tapeworm, however you want to call it. Then we have a 6-year-old, see, another kid. They're so gross. 6-year-old boy was taken to a local clinic for abdominal discomfort. A stool specimen was collected in formalin for routine ova and parasite examination. A and B show you what was observed in an unstained wet mount prep, and you have to tell me what it is. And really, we're trying to tell if this is Hymenolopis. I'm going to give you that, spoiler alert. But is it Hymenolopis nana, or Hymenolopis diminita? How can you tell them apart? Maybe pause, take a look at your table, or look at the book, or whatever you need to do, and then determine which one it is. And I'll tell you, the picture on the right is really the giveaway. And although you cannot necessarily judge the size, it does say it's between 40 and 40 micrometers. This is Hymenolopis nana. And that is nana as in nanometer. Remember Endolimax, that little one that haunted you? It is a smaller egg than Hymenolopis diminuta. We'll talk a little bit about that. It's usually more 70 to 85 micrometers versus the small 40 to 43. But what really gives you away what this is are these little filaments that run from one end of the egg to the other, one end of the egg to the other. And they are called polar filaments because this is a pole, you know, like a top, and this is a pole, like a bottom. And diminuta does not have those. So there are no polar filaments in Hymenolopis diminuta. If we look at the way that Hymenolopis nana looks, it has that armored, very sticky outy rostellum. It does have 4 suckers, really short and fat little proglottids. It has a short row of hooks on the protruding rostellum, and the proglottids are wider rather than they are long. So even in Diphyllobothrium latum, these are even shorter. I wouldn't say they're fatter, but they're definitely smaller than Diphyllobothrium latum was. This, I feel like this is like such a sad picture. Doesn't this little critter look so sad? I don't know, but this is a scanning electron micrograph of Hymenolopis nana. And this is affectionately known as the rat tapeworm. You can see here that humans and rodents are infected when they ingest the cysticercoid infected arthropod. It actually gets into some sort of insect that, again, usually by accident, we ingest. And once it gets into us, you can see that the oncosphere will hatch. And the... Oh, let me backtrack here. You can ingest the cysticercoid arthropod, or you can ingest the egg as well. So either one of those things are kind of gonna lend themselves to the same result. Basically, I was jumping ahead and it did not make sense. You have this oncosphere hatching, which is coming out of this egg, and the cysticercus from the flea getting into the intestinal villi and, getting through and attaching. It develops into an adult. The adult is actually in that later portion or ileal portion of the small intestine. And then, as you saw with other ones, the eggs are released to the genital atrium of the gravid proglottids, also known as the genital pores. The gravid proglottids can also disintegrate and release eggs that are passed in stool, and then the human will pass the eggs in the feces, which is then eaten by a bug, which is eaten by a rat, which it poops and we eat an egg, or we actually get the cysticercoid larva. But I'm guessing that probably the egg is more common, what we would ingest. And last but not least, we have Hymenolopis diminuta. You can see that the egg has some thickenings around the side. It does not have polar filaments, so that's important. It does have an oncosphere. There's no rostellum, so it's just got 4 suckers and no rostellum. Here's a sad picture of this little dude. I just don't know why they look so sad to me. But either way, the Scolex has no rostellum at all, but it does have 4 suckers, and it has pretty similar proglottids to the Hymenolopis nana. All of these things that I said here. This is a cool, I really like scanning electron pictures. So this is a cool picture of eggs sort of just around in the intestinal wall or stuck in something anyway. And very similar to Nana, you have rodents being infected oftentimes in place of humans or at the same time. And if we look here, there's an egg that's passed in the feces, no polar filaments. It's ingested by an arthropod intermediate host, which then hatches the oncosphere, which penetrates the intestinal wall of the arthropod, which then develops into the cysticerci in the body cavity of the insect. And the bug is eaten by a rat or a human, and once it's eaten, that little cysticercus will then not hatch, it's not really enclosed in anything, will attach to the small intestine, just like we saw in Hymenolopis nana. It will shed proglottids, shed eggs, and they will be passed just as we have seen before. This is the last show, or the last video in the series. Make sure you let me know if you have any questions or if I can help.